Hi there. In this final video of this section, we'll talk about how cells interact with each other via cadherin proteins and how they interact with the extracellular matrix using integrin proteins. This slide summarizes a lot about those interactions. We'll focus in particular about cell-cell contacts at the adherens junctions and the desmosome. These are cadherin proteins interacting with each other from each cell. We'll talk about how these interaction points interface with the cell internal cytoskeletal elements, intermediate filaments connecting to the cadherins at the desmosome, and actin rings in cells interacting at adherens junctions. The integrin proteins that we're going to talk about are where the cells interact with the basal lamina, which is also called the basement membrane. This is a collagen-based structure that has a lot of tensile strength and serves as a major barrier between the outside world, where the epithelial cells considered are, and then the inside world of your body with connective tissue and fibroblasts. We won't say too much about tight junctions, though they also interact with the actin network and form a strong impermeable barrier between cells, preventing solutes from diffusing into the space in between cells. Gap junctions actually open up channels between the two cells, so there's some communication between the cytoplasm of this cell and its nearest neighbor. Here we just are labeling some of the parts of the epithelial cell. Again, we've talked about the apical edges. We've talked about cell polarity, how microtubules end up producing an asymmetric position for the nucleus, the basal side of the epithelial cell, the basal lamina, also the basement membrane, and then an extracellular matrix that exists underneath this. I sometimes talk about this as the barrier between the inside and the outside world, and that sort of terminology that comes from cancer. Epithelial cells are the source of 85% of all solid tumors, as long as they are maintained out here, or they produce growth that extend out into the lumen, they will be considered localized. It's when they break through the basal lamina and grow on into the inside that tumors are considered to be invasive, and then they're at high risk for metastasis by spreading through the bloodstream that exists down here. Cadherin proteins are the types of proteins that interact with each other, that tie cells together. They interact with the intermediate filaments and also the actin filaments on the inside of the cell. They are molecule specific for a tissue type, since they also bind calcium and they adhere to each other. That's the source of their name, cadherins. Homophilic binding refers to the fact that these two molecules are the same, even though they're different colors. Because homophilic interactions match up cells of a particular type, you can have mixtures of cells and arising from two different tissues, and these can then sort themselves out through self-association with the types of cells that they normally interact with for producing different tissue types. They can also sort even identical cadherins based on amounts that they can have molecules that are supposed to be on the inside of a tissue versus the outside, all based on the thermodynamics of the interactions between these proteins. So here we see an attempt at a sort of a 3D picture of this, what's called the adhesion belt, a ring of actin surrounding the top of, near the apex of the epithelial cells. These rings are connected to each other via the cadherins at the adherens junctions, and it's this belt that also gives rise to the actin bundles, which extend from this going up into the microvilli, providing them with their characteristic stiffness. Desmosomes, on the other hand, are not rings of bands of intermediate filaments are connected in there, but they're sort of spot welds is the same. These are different cadherins from the ones used at the adherens junctions, though they still self-associate with a homophilic partner. It's the cytoplasmic ends of these cadherins that are more different in that they interact with the intermediate filaments. but they are, as we've mentioned, they're very important for the integrity of the sheet of epithelial cells as a whole. Desmosomes involve attachments of two cells, and hemidesmosomes mediate the interaction of one cell to the extracellular matrix. Here are the proteins which are connecting the intermediate fillets of the internal cytoskeleton to the outside world are proteins called integrins, and this is a large family of proteins. And it's these hemidesmosomes that provide the attachment points to the extracellular matrix, or the surface on which the cell is sitting. The last point is that integrins not only connect to the intermediate filaments, but they also connect to signaling pathways, such that these molecules serve as anchors but also as sensors for the extracellular environment, which are then responsible for transmitting the connections to the inside of the cell, which responds by the transcription of genes appropriate for the current attachment sites. This slide pretty much summarizes the entire material covered in this lecture. The 5 types of polymers that we've talked about today: 3 cytoskeletal elements, primarily for the internal cytoskeleton, 2 types of proteins that interact with external interactions, the adherence junctions interacting with actin, the desmosomes and hemidesmosomes interacting with intermediate filaments, and these molecules all give rise to the structural integrity of the cell and the tissue. On this final slide, I've just tried to summarize some of the points covered in a relatively succinct way. Okay, thank you for listening.